Hello students, welcome to EPG Pathshala. I am Dr. Pooja Gulati from Department of Microbiology, Maharishi Dayanand University, Rohtak. Today we are going to talk on the module Immunodiagnostics under the paper Animal Cell Biotechnology. After studying this module, the students, you will be acquainted with basic knowledge of the antigen-antibody interaction, the methodology and the principle behind various immunological methods, the application of these methods for detection of pathogen, and the advantages and disadvantages of various diagnostic methods. To start with, presently the immunodiagnostics have evolved over a past few years, and diagnostics have reached a new height because of these immunodiagnostic method. The immunodiagnostic methods are important methods which are based upon antigen-antibody interaction. As we know, the antigen and antibodies are highly specific reactions similar to an enzyme-substrate reaction, but the only difference between enzyme-substrate and the antigen-antibody interaction is that in the case of enzyme substrate reaction, the substrate is converted into the product, but after the antigen antibody interaction, which is as specific as enzyme substrate reaction, there is no change in the antigen or the antibody after the reaction. So the immunoassays are basically culture independent methods which are based on antigen antibody reactions. So students, before we proceed to various immunological methods which are commonly used in the laboratory, I will tell you about some of the advantages of these methods which have really allowed these methods to be included in each and every clinical laboratory. These immunological methods firstly allow early, economical and rapid diagnosis of several diseases. Then secondly, the most important thing is that they provide a real-time information about the disease and their treatment response if quantification of the pathogen is carried out. And third, and the most important, these techniques, they serve as a gold standard when biological sample or the tissue specimen is not available for uncultivable organisms and for asymptomatic carriers of the disease. We will discuss different immunological reactions. All these reactions can be performed in several formats like precipitation, agglutination, complement fixation, radioimmunoassay, ELISA, then chemiluminescent immunoassay, immunofluorescence, and immunoelectron microscopy. So first of all, we will discuss precipitation reaction. Precipitation reaction involves a soluble antigen and antibody, and this reaction can take place in different formats, but it has to be occurring in optimal conditions in the presence of electrolytes, which leads to the formation of insoluble precipitate. As you can see in figure 1, when antigen and antibody meet, then when optimum concentration of the antigen and antibody are present, in that concentration, a lattice-like structure is formed. This lattice-like structure results in formation of an precipitate. So when the antibody is present in higher amounts, as seen in the blue section of the figure, then also the lattice formation will not take place, and this effect is known as Prozon effect. Also, if antigen is present in excess, all the Fc portions of the antibodies will be occupied by the antigen and no precipitate will be formed. This forms the principle of all the precipitation reaction. The precipitation reactions can be performed in different formats. The different formats in which the precipitation reactions can be performed. The precipitation reactions can be performed in liquid-based media or gel-based media or immunoelectrophoresis. Further, liquid-based media are divided into ring tests, light tests. Gel-based is divided into radial immunoassay, double diffusion in 2 dimensions, immunoelectrophoresis into countercurrent immunoelectrophoresis, and rocket immunoelectrophoresis. In the coming slides, we will discuss each of these patterns. liquid media-based precipitation reaction. These can be performed in tubes, that is ring test, or on a slide, that is slide test. In the tube, the soluble antigen is layered over a column of antiserum in a tube, and the tube is allowed to stand for some time. The ring of precipitate is formed at the junction of these two liquids. This test is very easy to perform, economical, and is commonly used in the laboratories, especially in the Escoli's thermoprecipitation reaction and in Lancy field technique used for grouping streptococci. Secondly, we can perform this test on the slide. Antigen and antisera are mixed with each other on the slide and observed for the formation of floccules. This test forms the basis of VDRL tests used commonly in the detection and diagnosis of syphilis. Many other pathogens can be detected in a similar way. Here we discuss gel-based precipitation reactions. These reactions are also known as immunodiffusion reactions. The radial immunodiffusion reaction was first discovered by Mancini, and thus it is known as Mancini's method. Antibodies are mixed in the agar gel and poured onto the glass slide or Petri plate, as you can see in the figure. Wells are cut on the surface of the gel and antigen is loaded into them. The antigen will diffuse radially and form concentric ring of precipitation. The concentration of the antigen can be estimated by the diameter of the halo. Thus, in this test, we can also is based for both qualitative and quantitative estimation. This test is commonly used for detection of influenza virus. We'll discuss double immunodiffusion in 2 dimensions, and this method is commonly known as Octoloni method. In this method, various antigens and antisera can be compared. Wells are made on agar slide and the antiserum is placed in the center well. Antigens are present in the surrounding well. The pattern line of precipitate is used to determine the relatedness of different antigens. If the two antigens are related, then fused pattern will be obtained or fused precipitation line will be obtained. If they are not related, then cross lines will be obtained. And if partial identity is observed, that is identity of some of the epitopes of the antigen, not all, then a spur formation will be observed as shown in the figure. Here we will discuss immunoelectrophoresis. This technique was first developed by Graber and Williams, and it is a blend of electrophoretic and immunodiffusion method. In this technique, the antigen is placed on the well of an agar or agarose gel and is electrophoresed for an hour for the separation of its constituent proteins. So in this case, a mixture is poured into the well which will separate into different components depending upon its ionic concentration. The antibody is placed in the trough which is cut on agar gel above the antigen well, and diffusion takes place for 18 to 24 hours. Different precipitation lines are observed for different proteins. If the antisera will contain the antibodies against the protein, then the line will be observed, otherwise it will not be observed, and thus we can tell the separated protein mixture contains how many different types of antigens. Now let me introduce you to countercurrent immunoelectrophoresis, which is a common type of immunoelectrophoresis. In this technique, on a slide, a gel of agarose is poured, and on two ends, two wells are created. In one end, antigen is placed, and the other end, antibody is placed, and they're allowed to diffuse overnight. And a line of precipitation can be seen in between on the diffusion wherever there is an equivalence zone. This technique, if is driven electrically, then it is known as immunoelectrophoresis. So it is different from the normal electrophoresis in the method that when this is driven by the electricity, the movement will be fast and the precipitation line can be formed much earlier, within half an hour, and thus this technique is 10 times faster than the conventional double diffusion method. This technique has several applications and it is employed for the detection of phytoprotein in serum and cryptococcus and meningococcus in cerebrospinal fluid. Here we discuss different types of immunoelectrophoresis. One we have already discussed. Coming on to rocket immunoelectrophoresis, it is both qualitative and quantitative method. The antisera against the test antigen is incorporated into the agar-rose gel and poured onto the slide. Antigen are loaded in a series of well in the gel in the increasing concentration as shown in the figure. Electrophoresis will take place and rocket-like precipitation lines are formed. The height of the precipitation line or the rocket will increase with the increasing concentration of the antigen, and the area under the rocket can be used to quantitate the amount of antigen by preparing a standard curve. Agglutination reactions. These reactions are similar to precipitation reactions, but the basic difference is the antigens here insoluble, that is they are bound to a carrier, but in precipitation reaction the antigens are soluble. Agglutination techniques are more sensitive as compared to the precipitation reaction because here we can observe the results immediately after the test is performed. Agglutination reaction, like precipitation, can also be performed in different formats like slide agglutination, tube agglutination, heterophile agglutination, agglutination inhibition, passive agglutination, and we will discuss each of them in the upcoming slides. So students, let me discuss now types of agglutination reaction, and the agglutination reaction which is commonly used is a slide agglutination reaction. In this, a uniform suspension of the antigen is made in a drop of saline and a drop of antiserum is poured over it. Now, if the agglutination occurs, that is clumping of the particle is seen, then the test is positive, and if the drop is clear without any clumping, then it is negative. This technique is commonly used in the laboratories for blood grouping, cross matching, and identification of bacteria from clinical specimen. Now, in addition to the slide agglutination, the tube agglutination test can also be used. The same test, but it is carried out in a tube and it is used for quantification of antibodies. Here, a fixed volume of the antigen is added to equal volume of serial twofold serial dilutions of the antiserum. And the highest dilution in which the agglutination occurs is recorded as the antibody titer of the serum. And thus, in this method, we can quantitate the amount of the antibody which is present in the serum. This test is commonly used for diagnosis of typhoid, and as you all must be knowing that typhoid, the most common test is Vidal, and Vidal test is based upon agglutination reaction. Then it is used for detection of brucellosis and typhus fever. In passive agglutination test, we use the principle of agglutination, that is insoluble antigen is bound to an antibody. Whereas in precipitation, our antigen was soluble. So if we want to make a soluble antigen into an insoluble antigen or a particulate antigen, then we can link it to a carrier molecule like latex, RBC or bentonite, and thus an antigen which is present in the soluble form can be fixed onto the carrier and can become particulate antigen, and thus agglutination can be performed. So what is the advantages of this method is that agglutination can be easily seen with the eyes and is a faster method as compared to the precipitation. As a carrier, we can use RBCs. If RBCs is used as a carrier, then this method is known as hemagglutination test. If polystyrene latex beads are used, then this method is called as latex agglutination test. This test is highly important, is commonly used for detection of hepatitis B surface antigen, RA factor, rheumatoid arthritis factor, in bacterial typing, especially of Neisseria meningitis, and for detection of many other soluble antigens. Another modification of this method is reverse passive agglutination test or co-agglutination test. In this method, the antibodies are attached to the carrier surface rather than the antigen. And in order to attach the antigen, a carrier molecule which is used is a strain of Staphylococcus aureus which contains protein A. This Staphylococcus has a protein A on its surface which will bind to the antibody and will link to it, and thus will immobilize the antibody on the surface of the carrier. The Fc portion of the IgG binds to the protein A, and thus the Fab fragments are freely available to bind to the test antigen, resulting in the agglutination. So reverse passive agglutination is just opposite of the passive agglutination. In passive agglutination, we were detecting antigen and soluble antigen was made fixed, and here the antibodies are fixed on the carrier particle, which is actually protein A. A modification of the passive agglutination is reverse passive agglutination or co-agglutination method, and in this method, instead of the antigen, antibody is attached to the surface of the carrier. And to attach the antibody, we use a carrier which is actually a strain of Staphylococcus aureus containing protein A on its surface. This protein A has an ability to bind to the Fc portion of IgG, and thus the Fab fragment is thus free to bind to any test antigen, resulting in agglutination. So in this method, we are actually detecting antigen and we are fixing the antibody on the surface of the carrier protein. This method has found several applications in the clinical laboratory. For example, it is used for the detection of antigen in urine, blood, and cerebrospinal fluid. It is used for detection of Neisseria, Streptococcus, and Haemophilus. Now, another modification of heme agglutination is heme agglutination inhibition test. In this, the patient's serum antibody is first incubated with the live hemagglutinin virus. So in heme agglutination inhibition, we first incubate the test serum or the patient serum with the live hemagglutinating virus. So hemagglutinating virus is a virus which has an inherent ability to bind to the RBCs. So if the patient serum does not contain the antibodies, then the antibodies cannot bind to the virus surface. Antibodies which are specific to the virus, then it will not bind to the virus, and the viruses will agglutinate the RBCs. It means if the agglutination has occurred, there is no specific antibody in the patient's serum to that particular virus, indicating a negative agglutination as a positive result. On the other hand, if the patient's serum lacks virus-specific antibodies, then they will not bind to the agglutinating virus. If they will not bind to the agglutinating virus, then virus is available freely for the RBCs to agglutinate, and agglutination will occur. And thus, in this method, lesser the agglutination, more is the viral load present in the patient. Now, this method has several applications. For example, it is used in detection of viral infections, which has ability to bind to RBCs like rubella and influenza. Here we discuss complement fixation test. This test is very important test and is based upon the principle that antigen and antibody complexes can bind or fix the complement. This system cannot be visualized without an indicator system. In this method, the indicators are the sheep erythrocytes, which are coated by the rabbit antibodies, which can easily lyse the complement system. So, if the antibodies are present in the test serum, they are first incubated with the antigen and later with the complement. If the antibodies are present in the test serum, it will bind to the antigen and the complement will be fixed and will not be available to lyse the indicator system, that is sheep erythrocytes coated with the rabbit antibodies, that is your AMBO receptor. Thus, no hemagglutination will take place. So from this we infer that if the antibodies are present in the serum against the test antigen, they will fix the complement and sensitize sheep RBCs will not be lysed. Here the negative result is actually the positive result, because visualization of hemagglutination shows that test serum does not contain antibody. Thus, antigen-antibody complexes are not formed, complement is not fixed, and can cause the lysis of the sheep erythrocytes. This method is very important method and has various applications. It is used in the Weizmann reaction for diagnosis of syphilis, immobilization test for diagnosis of Treponema pallidum, then immune adherence for detection of Vibrio cholera, cytolytic or cytosol test for detection of Vibrio cholera. So here we will discuss radioimmunoassays. The radioimmunoassays were first developed by Burson and Yellow in the year 1959, and for this they received a Nobel Prize in 1977. Only Yellow received the Nobel Prize as the Burson due to the Burson's death. The principle behind this technique is competitive binding of radio-labeled antigen and unlabeled antigen to the high affinity antibody. This method has several applications in the field of medicine and biology. For example, the quantitative analysis of IgE, which is responsible for allergic reactions, viral antigens, drugs, hormones, tumor markers, etc. And the major limitation of this method is that this method relies on the use of radioactive material. So this method forms the basis for the other immunoassays, especially ELISA, which is enzyme-linked radioassay, which is based upon the same principle, but here the radioactive substances are not used. I will briefly discuss the mechanism or the protocol of RIA. In this method, the labeled antigen is mixed with the antibody at a concentration that saturates the antigen binding sites of the antibody. So first of all, as shown in the figure, the antibodies are fixed onto a well or a tube. Later, the labeled antigen which is radio-labeled antigen is added at a concentration which will saturate all the antigen binding sites of the antibody. After incubation, a washing is done to remove the unattached labeled antigen. Following this, unlabeled test sample are added in a progressively larger amounts. The unlabeled and the labeled antigen will compete for the available binding sites on the antibody. The progressive increase in the unlabeled antigen displaces the labeled antigen from the binding site. Thus, there is a decrease in the amount of radioactivity of antigen-antibody complex, which can be measured by precipitation of the complex. The amount of the test antigen is determined using the standard curve. So this method is also a quantitative method, as the amount of the antigen which is present in the test serum can be quantitated. So ELISA has the same principle as RIA. It differs from the RIA in the detection method. Detection here is not based on radio-labeled antigen. It is based upon the enzyme catalyzed reaction or fluorescent probe. For example, here we use the labels like horseradish peroxidases, alkaline phosphatases, glucose oxidases. This method can be performed in different formats. So there are different types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. As you can see in the figure, direct ELISA is used for the detection of the antigen directly, that is those antigens which are present in a tissue or which are viral samples, so that viral samples can be directly detected. In this case, the antigen, the bound antigen is incubated with the primary antibody, and this antibody is conjugated with the enzyme like I have discussed, horseradish peroxidases. After incubation for a short period of time, washing is done. The unlabeled antibody will be removed. If the antibody is specific to the antigen, it will bound to the antigen, result in the binding of the enzyme. So later we add the substrate for the enzyme. The substrate used here is a chromogenic substrate, that is which when hydrolyzed by the enzyme or cleaved by the enzyme into the product can be seen with the eyes. So it is chromogenic, so it will lead or give rise to a colored end product. I will repeat again that if antigen is present, it will bound to the labeled antibody. The antibody will further break down the substrate into a colored product which can be easily detected either visually or by means of special instruments like ELISA reader. Then indirect ELISA is more commonly used method, but here we are detecting the antibody in the test serum rather than the antigen. So on the microtiter plate, antigen is fixed to this fixed antigen, test serum is added. If the serum has specific antibodies, then they will bind to the antigen, and the secondary antibody which is labeled with the enzyme is added, which binds to the Fc portion of the bound antibody. The secondary antibody results in the binding of the enzyme. Later washing is done and again the substrate is added. Substrate is chromogenic. If the enzyme is present, the substrate will be converted into the colored product as in the direct ELISA. Then another modification is sandwich ELISA. In this case, we can again detect the antigen. So the capture antibody is fixed onto the microtiter plate. The sample which contains the antigen is later added. If the antigen is specific, it will bind to the antibody. If the antigen has bound, then we use another secondary antibody to another epitope of the antigen as the indicator antibody, and this antibody is labeled with the enzyme and will break down the chromogenic substrate into a colored product. Another variation of ELISA is competitive ELISA. It has same principle as the RIA. Here, first of all, the antigen is incubated with an antibody. If the antigen is specific, then all the antibody will be bound to the antigen, and upon adding it to a well which is coated with the same antigen, if the antibody has some free sites available, then only it can bind to the fixed antigen, and thus the amount of antigen in the serum will leave less of the amount of the antigen in the serum will leave less amount of the free antibodies to bind to the well antigen, as there will be competition for binding of the Fab fragments or the antigen binding fragment of the antibody, and the color can be detected in similar methods. But here the results will be more the amount of the color produced, it means less the amount of the antigen in the serum. Now, ELISA has many applications. ELISA is the most common immunological technique that is used to detect diseases. Some of the applications of ELISA are detection of infectious diseases like dengue, influenza, hepatitis, Epstein-Barr virus. Then there is we can detect viruses like HIV. Then we can detect the titer of antibodies which are produced in the patients of TB, that is mycobacterial antibodies. Then in addition to this, important viruses like rotavirus can also be detected by ELISA. Even E. coli can be detected in the fecal samples using ELISA. Now, in addition to the detection of the pathogens, various toxins which are present in the foods like penicillins, aflatoxins, and food adulterants can also be detected by means of ELISA. Even though the ELISA has so many applications, still it cannot reach every laboratory, as we need an instrument, that is ELISA reader, which makes it not feasible for every laboratory. Thus, a modification of ELISA is formed, in which instead of the plastic plates which are used for ELISA, we use nitrocellulose or nylon paper strips. In this method, either the antigen or the antibody is blotted at one side of the strip in the form in the form of a dot, and this can be either an antigen or an antibody. If we are detecting the antibody, we blot the antigen. If we want to detect the antigen, we blot the antibody. And then all the detection steps are similar in the ELISA, that is, the binding can be detected by binding of another antibody, that is secondary antibody, which is labeled to the enzyme. And this secondary antibody the binding of the secondary antibody can be detected by means of adding a chromogenic substrate, which will give the colored reaction. The dot ELISA is commonly used has many applications and is commonly used in the laboratory for detection of amoebiasis, cryptosporidiosis, and many other infectious diseases. Chemiluminescence immunoassay. So first, what is chemiluminescence? Chemiluminescence is the phenomena of emission of energy in the form of light in some chemical reaction. Chemiluminescent compounds like luminol or acridine esters are used as the label in place of enzymes and radioisotopes, which were used in the ELISA and RIA. So these methods are similar to ELISA, just the substrate, instead of the chromogenic substrate, we are using the chemiluminescence substrate or the luxogenic substrate, which when converted into the product by the enzyme gives a light signal. This method is more sensitive. For example, oxidation of the compound luminol by H2O2 and enzyme Haus-Radix peroxidases produces light. This method can be fully automated and is used in the laboratory where large amount of the samples have to be handled. Immunofluorescence test. This test also has the same principle as ELISA and RIA, but here instead of tagging the antibody molecule with the enzyme or the radiolabel substrate, we use fluorescent dye or fluorochromes, which are detected by colored light emission when excited by light of appropriate wavelength. The emitted light can be viewed with the fluorescent microscope, which is equipped with UV light source. The commonly used dyes for this test are fluorescein, phycoerythrin, rhodamine, and so on, which are listed in this slide. This method is also a more sensitive method, can be easily automated, and is used for identification of bacteria, viruses, and other antigen. The method is available in two formats, that is direct immunofluorescence and indirect immunofluorescence. The direct immunofluorescence is similar as direct ELISA. The primary antibody is used, which is chemically linked to the fluorophore. This primary antibody will bind to the antigen. The fluorophore will facilitate the visualization of the target antigen under fluorescent microscope. Then indirect ELISA is similar as indirect immunofluorescence is similar to indirect ELISA. This method uses two antibodies, a primary antibody which is unlabeled and the secondary antibody which is labeled. The primary antibody binds to the antigen and the secondary antibody has the fluorophore dye which will bind to the primary antibody and will give the signal. Immunoelectron microscopy. In this technique, we can detect the antigens which are bound on the surface of the cells using an electron microscope. And so here an electron dense label is conjugated to the Fc portion of the specific antibody for direct staining, or conjugated to an antibody reagent for indirect staining. Electron dense label used here are ferritin and colloidal gold. These labels will absorb the electrons and can be visualized easily with the electron microscope as small black dots. It means that if we use different antibodies to different antigens present on the surface of one cell, then varying the size of the label, like gold particles of different size can be bound to different antibodies, and simultaneous detection of the antigen on the surface of cell can be performed. This is one of the very important advantage of immunoelectron microscopy. Now let us discuss what are the challenges of these immunodiagnostic techniques. The serodiagnostic techniques have for various parasitic diseases like African trypanosomiasis, amoebiasis, filariasis are not approved by FDA. So this approval has to be done before this technique is adopted in any laboratory. Secondly, optimization remains an issue for the immunodiagnostics as the results are easily affected by intrinsic and extrinsic factors like different antigenic preparations. They result in different results in different laboratory. Then in addition to this, different laboratories may use different matrices on which the antigen or the antibody is bound and which can result in different results. Then a major issue or the challenge with the immunodiagnostics is cross reaction. Cross reactions means if a parasite or if a pathogen has an antigen or antigen which is common with another pathogen, then we can get false positive or false positive results which are really harmful. So students, let us now summarize what we have learned in this module. So this module shows that the specificity and variety of antigen-antibody reaction have made the immunodiagnostics unique and have allowed fast, inexpensive, accurate diagnosis of several infectious diseases. Now, these techniques can be based on direct detection of the antigen, or we can detect the antibodies to these antigen, resulting in the detection of the various pathogens. Now, the techniques like latex agglutination, ELISA, immunofluorescence are actually preferred techniques which can be easily adopted by various laboratories for the accurate detection. Now, automation of these immunological methods can be done, which can revolutionize the field of diagnostics in laboratories and can increase the specificity and sensitivity of the detection methods many folds. Thank you.